All right, AP Bio, this is my video on science practices, which are the skills needed for any scientist, but you will be tested on these skills for your AP Biology exam. All right, so I'm going to go over scientific questioning and procedures or methods. This is basically how we conduct experiments in AP Biology. So we've done this many times. The first thing I have you guys do is come up with a testable question. So based on your prior knowledge and observations, you come up with a question that you want to test. Okay, once you've decided on the question, you then form a hypothesis, which is your prediction based on your understanding of biology. Not to be confused with the null hypothesis, which is a hypothesis you form for your chi-square analysis. Remember, null means no difference. So this hypothesis by default states that there's no difference between the two groups that you're testing. And then you set up the procedures. A valid protocol would include a control group to compare your experimental group to. So I'm going to set up a control group and an experimental group. And it's really important that when you design your experiment that you control for all variables as much as possible. I mean, there's some things that are out of our control, but whatever you can control, you want to make sure those variables are the same for both groups. So for example, if I'm testing the effect of temperature on cellular respiration of germinating seeds, you got to make sure that all other variables besides temperature are the same for those groups. For the experimental group, you have to then add a variable that you are testing. So if you're testing the effect of temperature, you're only going to add that variable to the experimental group. The control group could be your room temperature, for example, and then you change the experimental group to either really hot temperature or cold temperature. Okay, that's known as your independent variable. Okay, that's the variable that you're manipulating, the variable that you are changing. How will you measure the effect of the variable? You will have to collect dependent variable data. Okay, so the dependent variable depends on the manipulated variable or independent variable. It's really important that you define what that variable is. How will you know what the effect of the independent variable is? So in the case of cellular respiration, your dependent variable could be the amount of CO2 that's released, for example. So the more CO2 is released, the more cellular respiration is going on. Now, when you're graphing your data, keep in mind your independent variable is usually on the x-axis, and the dependent variable will go on the y-axis. Don't forget to include units of measurement. Okay, let's do a quick review on the different types of graphs that we have been using. Anytime you're correlating two variables, so you're looking at how one variable affects another, we use a scatter plot. Okay, scatter plot is just dots, okay, on the x-axis correlating to the y-axis. Okay, when you are comparing groups or different variables, for example, you're testing different types of fertilizers, you will then use a bar graph. Remember to keep space between your bars. Anytime you're representing a range or distribution within a population, remember that's a histogram, and the bar graphs are attached. There's no spaces. Histogram. Okay, a lot of times we'll be looking at change over time where time is on the x-axis. In that case, you can use a line graph. Okay, so once you've set up the experiment and you've tested your variable, the other thing I want to emphasize, in order for you to get valid results, it's important to think about the number of subjects, the number of subjects that you're testing the variable on. And in science, it's always a good idea to increase the number of subjects as much as is possible. Okay, why is that? Remember, one of the statistical tests that we covered in AP Bio is standard deviation, represented by S, and standard error of the mean. So the standard error of the mean is equal to the standard deviation over the square root of n. Let's do a quick review of standard deviation. Remember, standard deviation is basically the variation from the mean. So let's say you just took a test and this is the mean. These are the number of students that receive different scores. And then there's another test with a mean, but a broader curve. So remember, the wider the curve, the larger the standard deviation, the greater the variation around the mean. Okay, the smaller the standard deviation, the less variation. Okay, by definition, one standard deviation includes 68% of a sample. You don't have to memorize this number. Just understand that if you include a greater standard deviation, like 2 standard deviation, it covers more of the sample, so 95% of the sample. So in AP Bio, on free response questions, they'll often ask you to graph the data within 2 standard errors of the mean. Okay, and it's represented like this, 2 times SCX. And the reason that they ask you to graph within 2 standard errors of the mean is because this includes 95% of the sample. So it includes 2 standard deviations. Remember, 2 standard deviations covers 95% of the sample. In other words, you can have 95% confidence if you graph within 2 standard errors of the mean. So here's an example. Let's say we're looking at the height of a plant in centimeters. And we have different types of fertilizers that we're testing. So that's your independent variable. And because these are different groups, I can use a bar graph. And then the standard error of the mean is represented above and below the mean. So let's say your standard error, your one standard error, let's say is 1. Then your 2 standard error would be 2 times 1. So you would go 2 units above and 2 units below the mean. Okay, and in science, you don't want the standard error bars to overlap. Okay, it's always better if you want to show distinct differences if those standard errors do not overlap. All right, so that's the data and data analysis. In science, it's very important that your experiment is repeatable. So in the scientific community, a lot of times the work is published and read by your peers that are kind of studying the same problems. And if they repeat your design, they should get the same conclusion. If they don't, then you probably have to adjust your methods, think about next steps, new directions. So science is always changing and hopefully moving forward as you use other conclusions to kind of build upon that. Okay, the last thing I want to go over is chi-square. So in the beginning, I mentioned that there's a null hypothesis, which predicts that there's no difference between the 2 groups. Now, using your formula sheet, you should be able to calculate chi-square, and also you can find the critical values from the chi-square table. If your calculated chi-square value is greater than the critical value, then you can reject your null hypothesis. Okay, that means that the difference is not due to just chance or random events, but it's actually due to a factor. And the only factor, hopefully, that's different between the two groups is the variable that you're testing. So the chi-square analysis is, it supports that the observed effect is due to the variable that's being tested. So it's a very powerful statistical tool. All right, let's move on to another set of science practices. We call these models. Models are basically visual representations of a phenomenon. So phenomenon is anything that you observe in science. So models are important because they help us to understand what's going on. And there are many different types of models. I'll give you several examples. And then I'll also go over how the AP test wants you to use models. Probably the most famous model that we've covered is that of the DNA double helix. Remember, in 1953, Watson and Crick were awarded the Nobel Prize. Rosalind Franklin, Franklin's X-ray crystallography really helped them in figuring out the final double helical structure. So I'm adding her as well. Even though they weren't able to actually observe DNA, they came up with a model that matched the molecular structure. Okay, another example that we covered, another model, is that of an operon. We spend quite a bit of time on operons and how they're involved in gene regulation in prokaryotes. So just quickly, remember operons have a promoter region, an operator region, and then followed by genes that are related in some process. Okay, so that would be a visual representation of an operon. Okay, graphs can also serve as models. A lot of people don't remember that graphs are important models. So anytime you're being asked to create a model, keep in mind that you can produce a graph. So in evolution, for example, the industrial melanism phenomenon, where you have a population of light and dark moths with fewer dark moths, but over time, as the trees became darker, we found something called directional selection that selected the darker moths. So this would be a model, a graphical model representing what happened to the moth population. Okay, another example of a model is that of U-tubes. It's a picture showing a setup of a U-shaped tube with a selectively permeable membrane right in the middle and solutions of different concentrations, and you had to predict the movement of water over time. So in this case, water would go from the right to the left because there's more water on the right side in terms of relative concentration. There's less water on the left side, so water will diffuse to the left. Okay, so those are some examples of models. For the AP exam, be able to create models. So keep in mind, this could be a graph. This could be like a graphic organizer, a flow chart. For the electron transport chain, for example, you can draw the membrane and how electrons are used to push hydrogen ions into the intermembrane space, and then the hydrogen ions move back into the matrix to power ATP synthase. Remember, you can use a graph, you can draw a picture, and then explain that picture in the free response portion. Okay, oftentimes you're asked to describe models, so they will actually give you a picture or a diagram and have you interpret it or describe it. They can also ask you to make predictions if you change the model. And I saw quite a number of these types of questions in our cell communication unit. Remember, in the cell communication unit, we went over signal transduction pathways, and we specifically looked at that for insulin and how certain individuals have changes, slight alterations in the signal transduction pathway for insulin receptor, which can lead to diabetes. So you can be asked to describe a model or make predictions if that model changes. And then finally, you may also be asked to refine a model, which is similar to making a prediction. So in the DNA unit, I've seen many questions where they offer you a DNA sequence or part of a DNA sequence and have you transcribe that sequence into mRNA, and from mRNA, using the genetic code, predict the amino acid sequence. Oftentimes, they can ask you to refine that model after a mutation. So if a DNA mutation occurs, how would that change the amino acid or refine the amino acid sequence? So these are some examples of some of the models you might be asked to use.